Hi, my name is Annabelle Harris, and I'm a freshman at Clemson University, class of 2025. My major is biochemistry.
Hi, my name is Elsa Saine. I'm a freshman in the class of 2025 at Clemson University. My intended major is chemical engineering. This research project is a bioinformatics investigation of Excystation and the intestinal parasite Entamoeba histolytica. So what is E. histolytica? E. histolytica is a pathogenic amoeba that affects around 90 million people every year with severe diarrhea, nausea, stomach pain, and other symptoms for around 2 weeks. In some more extreme cases, E. histolytica can infect the bloodstream and cause a liver abscess, which can cause up to 100,000 deaths each year. However, those infected by E. histolytica only show symptoms 10% of the time. Unfortunately, the asymptomatic cases usually go untreated, which allows the disease cycle to continue when E. histolytica is passed out of the body. E. histolytica takes on two forms, cyst and trophozoite. In nature, E. histolytica will form cysts with tightened walls for protection. Once in its host organism, E. histolytica will begin to exist in the small intestine into its second form as a trophozoite, which is much more motile than the cyst. In this form, E. histolytica begins to invade and colonize the large intestine.
There's not a lot of information about what signals are actually causing these conversions in E. histolytica or what genes are involved in initiating these processes. So, in this project, we specifically studied genes involved in excystation. To do so, we used a large online database called AmoebaDB that contains extensive information about thousands of genes in different species of amoeba. AmoebaDB has been critical to our research in discovering possible genes involved in excystation. While our study is primarily about E. histolytica, there's not much information about genes in E. histolytica because of the difficulty of getting the organism to form cysts in a lab. Thus, we instead studied the close counterpart of E. histolytica, Entamoeba invadens, a parasite found in reptiles. On AmoebaDB, we searched for E. invadens genes with at least a 5-fold increase in transcription during either the end of encystation or the beginning of excystation, as well as removing hypothetical proteins which have unknown functions.
So though we found a lot of genes possibly involved in excystation, we will discuss a few of the most interesting which had substantial increases in TPM. The first is an E. invadens gene that encodes for sphingomyelinase. Sphingomyelin is a lipid that is essential for growth and encystation. We know this because when mycin is used to inhibit the synthesis of sphingolipids, trophozoites are unable to encysst. Therefore, sphingomyelinase, which breaks down sphingomyelin, is needed for excystation in order to deplete the amount of sphingolipids and thus prevent encystation of the trophozoites. Another is a gene in E. invadens that makes hexokinase. Hexokinase is one of the first enzymes involved in glycolysis, and therefore exerts a lot of control over glycolytic pathways. And because Entamoeba do not have mitochondria, glycolysis is the only process available for ATP production, and ATP is essential for trophozoite motility, growth, and pathogenesis. Next, we have a gene in E. invadens that encodes for RAC GTPase. The Rho family GTPases, the protein family that the RAC GTPases belong to, regulate the actin cytoskeleton. The Rho GTPase's ability to rearrange and reorganize the cytoskeleton is essential for trophozoite motility, pathogenesis, and invasion of the host. Another gene in E. invadens makes cysteine proteinase 1 precursor. Cysteine protease is an enzyme involved in the pathogenic processes of Entamoeba. It's needed to degrade the extracellular matrix of host cells, kill host cells, and break down host proteins. It has also been found that certain cysteine protease inhibitors are needed to stop excystation, which suggests that the CPs are needed to initiate excystation.
A gene that I looked at codes for ATPase AAA family protein. It's a gene that codes for many cellular activities such as protein folding, cytoskeletal regulation, organelle creation, and DNA replication. Some key functions are maintaining protein homeostasis, such as protein quality control mechanisms and autophagy of membranes and protein complexes. All of those processes are very important for cell growth, which makes sense that it would get switched on as a cell goes from a dormant-like state of being a cyst to the growing form of a trophocyte. Next is TP53 regulating kinase. It is essentially a tumor-suppressing gene that controls pro-autophagic and anti-autophagic function. That means that it controls both cellular growth and cellular death. When an Entamoeba excises, it breaks down its own chitin wall to become a trophocyte form. This gene is most likely turned on to help with that process of growth and breakdown. In conclusion, there are many genes involved in excystation, like the ones that we looked at today. They code for different things, but all with the same goal of excystation. There were several other genes that showed probable involvement in excystation, and this area definitely deserves more research, especially now that E. histolytica has been able to end cyst in a lab setting thanks to Dr. Ingram-Smith's lab. We would like to thank everyone in the EUREKA program, the Clemson Honors College, and Dr. Ingram-Smith for helping us with our research. Thank you.